Hello and welcome back to Anthropology 1510. Last time we began Lecture 4 and we were talking about evolutionary theory. And so we're going to pick up from there and move into a discussion of genetics at this point. So let's get started. Last time we wrapped up our discussion of evolutionary theory by talking about Charles Darwin and how he published On the Origin of Species by Means of Natural Selection in 1859. And while a debate did erupt, from a scientific perspective, the evidence that comes in continues to support his ideas. Of course, as we said, he didn't have all the answers, and one of the things he didn't understand is how biological inheritance worked. He knew that offspring tended to resemble the parents, but he wasn't sure how those traits were passed down. You recall we talked about that theory of pangenesis, where they thought particles were being passed down, but they weren't sure exactly how that worked. They thought it all blended together in the offspring. Well, there was someone else who was working on this idea, trying to figure out how biological inheritance worked, and that's Gregor Mendel, who was an Augustinian monk. Mendel was interested in biological inheritance and trying to figure out what the mechanism was for passing down traits from parents to offspring. But he was also interested in using a scientific approach. That is, he was going to develop hypotheses and conduct experiments, that whole process that we talked about back in our scientific method lecture. In science, it's always a good idea to simplify your experiment as much as possible. That way it makes your conclusions more clear-cut. And so what Mendel wanted to do was find something that would reproduce fairly quickly. That is, it would produce offspring fairly quickly, so he could observe the passing of these traits from parent to offspring. He also wanted to find something that had a number of different traits he could examine, but for which the variation in these traits was very small. He settled on the common garden pea as a subject of study because it would reproduce fairly quickly. Plus, there were a number of traits for which there were only two variants, that is, there were only two possibilities. The color of the peas, for example. They were either green peas or they were yellow peas. When the plants were mature, you had some plants that were tall, you had some plants that were short. The surface texture of the peas, you had some that were rough and some that were smooth. So for a number of different traits, 14 of these different traits, there were only two variants, and so this would allow him to study how these two variants were passed down from parent to offspring in a simplified manner. So what he began to do first is to observe one trait at a time. For example, the height of the mature plant. Now there were some plants that were always tall when they were mature, and all they produced were tall plants. There were other plants that were always short when they were mature, and they always produced short plants. So you had tall plants, you had short plants. So he wondered, what would happen if I crossed tall plants with short plants? And what he found was, in that first offspring generation, all the plants were tall. There weren't any short plants, there weren't any in-between plants. Now, if you think about it, back when we were talking about pangenesis, if pangenesis were correct, then you'd expect these traits to be blended so that the height of the plants would be somewhere in between. But instead, they were all tall, just like that original plant had been tall, and none were short, and none were in between. Now, of course, he was going to have to explain how one trait could disappear completely, because no matter which trait he was looking at, whether it was the color of the peas, or the height of the plants, or the surface texture of the peas, always in that first offspring generation, one of the traits would disappear entirely. But he continued his experiment further. He crossed that first offspring generation. He would cross plants in that generation with one another. And what he discovered was, in that second offspring generation, that trait that had disappeared would reappear. Not some blended form like you'd expect with pangenesis, but it would appear just like it had been in that original parent population. And not only that, it would appear in a set ratio. For every 3 plants that exhibited the trait that was shown in that first offspring generation, there was 1 plant that would exhibit the trait that had disappeared entirely. And when we're talking about this, we have to remember that we're not talking about just a few plants like we have illustrated here. He's crossing hundreds of plants. He is studying thousands of peas. And he's seeing this disappearance of a trait in the first offspring generation. He's seeing the reappearance of the disappeared trait, of the trait that vanished entirely in that second offspring generation, in this 3-to-1 ratio. So how could he explain this? Well, we've already mentioned that they like to propose laws back in those days. And to explain the disappearance of that trait and the reappearance of the same trait, he proposed the law of segregation. Now, in the law of segregation, he states that each parent possesses 2 particles for a given trait. And then each parent only passes down one of those particles for a given trait to the offspring. It's not a situation as would have been predicted with pangenesis where one parent can pass down more particles than the other. What Mendel is saying here is that each parent possesses 2 particles for that trait, but is only going to give one to its offspring. And the only way that one trait could disappear entirely and then reappear exactly as it had been before is that the particles weren't blending. Now you recall pangenesis said things were going to blend all together in some unpredictable fashion, but he's saying no, that's not happening. These particles are not blending in the offspring. They're remaining segregated from one another. They're remaining separate from one another. He also proposed that one particle could be dominant. So if it was passed down, this dominant particle was passed down from parent to offspring, then that's what you would see. That's what would be exhibited in the offspring. And this is whether the offspring only received one dominant particle or if it received a dominant particle from both parents. The dominant particle is what you would see, the dominant trait. But the only way that you could have a trait disappear entirely and reappear is if there were also recessive particles. And with a recessive particle, the only way you'll see it is if each parent gives that offspring a recessive particle. If the dominant is present, if it's heterozygote, remember, if it's heterozygous for a trait, it has hetero meaning different, so it has one dominant, one recessive, then all you're going to see is the dominant. The only way you'll see that recessive trait is if both parents have given the offspring a recessive particle. So if it's homozygous recessive for that trait, then you'll see that. But let's look at our plants again, and that'll give us a better idea of how this works. Now, of course, these particles are what we would call genes today, but they weren't using that terminology. Mendel wasn't using that terminology at the time. He was still using particles. But if we go back and look at our plants, the capital letters in standard genetic terminology will stand as the dominant trait, and the lowercase letters will stand for the recessive trait. So in that original parent population, where you always had tall plants, always had short plants, then the tall plants would be homozygous dominant. That is, both particles they had would be dominant. In the plants that were always short, those are homozygous recessive, so that both particles that they have, for plant height in this case, are recessive. When you cross plants from that original parent population together, the one with 2 dominant particles can only pass down a dominant. The one with 2 recessive particles can only pass down a recessive particle. And so remember, the parent's only going to pass down one particle for the trait. So all of that next generation, that first offspring generation, every single one of those will possess one dominant particle, or one dominant gene is what we would call it, and one recessive. So they're all going to be heterozygous. So the dominant will be present in every single one of those plants. And so that recessive trait, at least as far as being able to see it goes, that recessive trait will disappear entirely. Now it'll still be there in the genetic makeup of the plant, but you won't be able to see it. The only thing you're going to see is that dominant trait. When we cross plants in that first offspring generation, when we crossbreed those together, now you've got these heterozygous plants. Each one has one dominant and one recessive. And so your possible outcomes for that second offspring generation are that each parent could pass down a dominant, so that would be the 2 capital T's. One parent could pass down a dominant and the other pass down a recessive, which would be heterozygous. The dominant would be present, so you'd see the dominant trait. Or the first parent could pass down a recessive and the second could pass down a dominant, so it's still heterozygous, the dominant is present, so that's what you see. Or both parents could pass down a recessive particle. And of course, if it's homozygous recessive, that's the trait that you'll see. You'll see that recessive trait. And that's why in that second offspring generation you wound up with that 3-to-1 ratio. For every 3 plants that exhibited the dominant trait, 1 plant exhibited the recessive trait. So this is important. He's saying these particles are being passed down. Each parent has 2 particles for a given trait. It's only passing down one of those, and the traits are not, these particles aren't blending together. They're remaining segregated. They're remaining separate from one another. And that's how this passing down of traits from parent to offspring works, at a basic level anyway. But his experiments weren't through. He also wanted to see what would happen if he followed 2 traits through this process at the same time. Would you still see the disappearance of a trait in that first offspring generation, and then the reappearance of that trait, or both traits actually, in that second offspring generation, in that 3-to-1 ratio? And so in looking at, in this example, the color of the peas and the surface texture of the peas, he had original parent populations in which plants would always produce yellow round peas. That's all that those plants would produce. You had other plants that would always produce green wrinkled peas. So you only had 2 variants for color. You had yellow and green. You only had 2 variants for the surface texture of the pea. You had either the round, the smooth, or you had wrinkled. So when you cross these together, what you'd expect to have happen by his law of segregation, you'd expect the dominant trait to be exhibited in that first offspring generation and the recessive trait to disappear entirely. And that's exactly what he found. He found that in that first offspring generation, the only peas that were produced were round yellow peas. There weren't any green peas produced. There weren't any wrinkled peas produced. The only thing produced were round yellow peas. So this tells him that yellow is dominant and round is dominant. Green is recessive and wrinkled is recessive. And so what he would expect to see in that second offspring generation is that 3-to-1 ratio, where you've got 3 yellows for every 1 green. You've got 3 round for every 1 wrinkled. And just as he predicted, that was the result. For every 3 plants producing yellow peas, there was 1 producing green. For every 3 plants producing round, there was 1 producing wrinkled. But he started to see new combinations that hadn't been seen anywhere before. Now you recall we started off with yellow round peas, or the plants that were producing yellow round peas, and plants that were producing green wrinkled peas. But now he started having plants producing green round peas and yellow wrinkled peas. So he was getting his 3-to-1 ratio for the yellow to green, and he was getting his 3-to-1 ratio for round to wrinkled. But now he's getting new combinations that had never even existed before. So how do you explain this? Well, he put some thought into it. And then he proposed his law of independent assortment to explain what was happening. Just as we saw with the law of segregation, each parent was passing on 1 particle for each trait, so that's what we would expect to have happen. But the particles for 1 trait would have to be inherited independently of particles for other traits if you have these new combinations. If they were tied to one another, you'd expect to always have the same outcome. So you'd expect there to always be yellow round or green wrinkled. You wouldn't expect to see any green round or yellow wrinkled peas. So those new combinations could only occur if the particles for 1 trait are inherited independently of particles for the other traits. So let's take a look and see what that looks like. So we're looking again at the peas that are being produced. You recall in that original parent population, what do we have? We have yellow round peas being produced, and we have green wrinkled peas being produced. And so when we cross plants from that original parent population together, what we expect to see is one of the variants for each trait to disappear entirely. And that's what we saw. We saw the green trait disappear entirely. We saw the wrinkled trait disappear entirely. All we saw was yellow round. Because you recall, all the plants in this generation are going to be heterozygous for this trait. They're going to have 1 dominant and 1 recessive, just like we saw with the law of segregation. The recessive will disappear entirely. We won't see it. It's going to be hidden and the only thing that will be exhibited will be that dominant trait. So there's nothing surprising here. We got the dominant traits exhibited and the recessive traits hidden. So in the second offspring generation, this is where the surprises came in, because we expect to see that 3-to-1 ratio of dominant to recessive, and that's what was exhibited. So that for every 3 plants producing yellow peas, you have 1 producing green. For every 3 producing round peas, you have 1 producing wrinkled. But then you have these new combinations. You have plants producing green round peas. You have plants producing yellow wrinkled peas. And so the way this is working, we know that the parent has 2 particles for a given trait. You can only pass down one of those particles to the offspring. Well, that entire first offspring generation is heterozygous, so each one of those plants can only pass down a dominant or a recessive, just like we saw in the law of segregation. But here's the key. The particle that it passes down for pea color is not connected to, it is independent from the particle that it passes down for the surface texture of the pea. So if that parent passes down the dominant for pea color, so it passes down that particle for yellow peas, it can still pass down either the particle for round peas or for wrinkled peas. And so since these traits are separated out independently from one another, this independent assortment, then we can have these new combinations. We can have these green round pea producing plants. We can have these wrinkled yellow pea producing plants. And so the important thing that Mendel did for us was move us away from that idea of blending inheritance like we saw with pangenesis, where inheritance would be some unpredictable result. But instead he's laying out the basics, saying that there are single particles passed down by each parent. The particles don't blend together in the offspring. So this is important. Remember this. It's a non-blending theory of inheritance. Things are remaining separate, segregated from one another. And that is what laid the foundation for our modern understanding of genetics. As we start to talk about genetics and the way we understand genetics to operate today, there are several terms that we need to be familiar with. Now, we'll talk about each one of these terms individually, but just so you know, I posted a file on Moodle that you can just download that has these definitions, so you don't have to sit and write all these down right now. Our first term is genetics, and by that we mean the scientific study of biological heredity. DNA, as we probably all know, at least if we've taken biology class, stands for deoxyribonucleic acid. But the important thing for us is that DNA carries the genetic blueprint for each organism. When we talk about a gene is a segment of DNA that carries the information, that carries the code for a particular trait. An allele is a form that a gene can take for a given trait. And when we're talking about a given trait, it may have several alleles. Now with Mendel's peas, for pea color for example, there were 2 alleles. There was yellow and there was green. For the surface texture of the pea, there was round and there was wrinkled. For some traits, there may be more than 2 alleles. The ABO blood group, for example, has 3 alleles. It has A, B, and O. So when we're talking about an allele, that is a form that a gene can take for a given trait. We've already mentioned the term homozygous, but homo meaning same. When we say something is homozygous for a trait, we mean it has identical alleles. It has the same allele for a given trait. So with Mendel's peas, if it had an allele, the dominant allele for pea color for yellow, from each parent, so that it had 2 of those, it's homozygous for that trait. And the same is true. It can be homozygous dominant in that case, or it can be homozygous recessive. If the individual inherited a recessive allele from each parent, it would be homozygous recessive. So homozygous, you can be either dominant or recessive, but it means you've got 2 dominants or 2 recessive alleles. If we say that an individual is heterozygous for a given trait, hetero meaning different, it means that the individual has different alleles for a given trait. And if we go back to Mendel's P's, that would mean it would have one dominant allele, one recessive allele. By genotype, we mean the genetic makeup of the individual. So this includes all the dominant alleles, all the recessive alleles, all the alleles that are present, all the genes that are present would be part of the genotypes. By phenotype, we mean the physical expression of the genes. Now, it's not going to tell you all the genes that are present like genotype will. It's just going to be what is exhibited, what you physically see. For example, with Mendel's P's, if you have a plant producing yellow peas, you can't tell by looking at those peas whether that plant has two dominant alleles for pea color or if it has one dominant and one recessive. So you can't tell if it's homozygous dominant or if it's heterozygous, because with the phenotype, all you're going to see is the expression of the genes. And finally, chromosomes are paired bodies that are made of DNA that actually physically carry that DNA from the parent to the offspring. So that's how this biological inheritance is transferred, by means of chromosomes. And we'll come back to chromosomes a little bit later in more detail, but I wanted you to be familiar with these terms at least a little bit now, so when they pop up you have some idea of what we're talking about. We're all on the same page with the definitions. And now we need to talk a little bit about how this information, this genetic information, is passed down from parent to offspring. This is not biology class, so we don't have a lot of time to spend on this. We're just going to hit some high points to get a general idea so we can move on and talk about how evolutionary theory, or how evolution works in conjunction with this to produce these changes over time. So we'll start out with the DNA. I think we all know the basic structure of DNA is that double helix, that twisted ladder. You got two sides of the ladder that are connected by rungs, and those rungs consist of nucleotides or bases. There are 4 bases. There's adenine, thymine, guanine, and cytosine. And those, when the DNA is being reproduced, combine in a certain way. Adenine always connects up with thymine. Guanine always connects up with cytosine when DNA is being reproduced. But what's important for us is that the sequence of bases along that strand of DNA code for amino acids. So every 3 base sequence will code for a specific amino acid. The amino acids are the building blocks of the proteins in turn are the building blocks for the organisms. So within this DNA code, that is where we have the blueprint for, that is where we have the information for constructing basically this organism, making it what it is. Within the cell we have 2 different types of DNA, both of which are important to us, but both are not important to us right now. As we discuss evolutionary theory and the passage of traits down from parent to offspring and how those can be modified over time, modifying the organism, bringing about that change over time, only one type is important to us. And that would be the nuclear DNA, the DNA that occurs inside the nucleus of the cell. The other type of DNA is mitochondrial DNA. And while it will be important to us later for tracing genetic lines, at this point, in terms of evolutionary theory, we're concerned with the nuclear DNA, because it's changes in the nuclear DNA that bring about evolutionary change. That's what will have evolutionary significance. So as we're thinking about this nuclear DNA, we need to think about cell division. And again, this is not biology class, so we don't have a lot of time to spend on this, but we do need to just touch on it just a little bit. The first type of cell division is mitosis. This is how all your body cells reproduce. And with mitosis, as cell division occurs, the DNA is replicated, and then where you have one parent cell, you have 2 daughter cells that are produced, and each of those daughter cells is a copy of that original parent cell. So each of the daughter cells has all the chromosomes, has all the genetic material that original parent cell had. So in terms of evolutionary theory and passing down traits, this is not important to us, this type of cell division, but this is how all the body cells are reproduced. The second type of cell division is meiosis. And this one is important to us because this is how that genetic information is passed down from parent to offspring. With meiosis, cell division actually occurs twice. The first time it occurs, the DNA replicates itself and the resulting cells have the full complement of DNA. That is, they've got all the chromosomes that the original parent cell had. But then cell division occurs again, and the result of that cell division is 4 daughter cells that each have half of the number of chromosomes that the original parent cell had. Now, these daughter cells would be the egg in human females, would be the sperm in human males. And when the egg and the sperm get together, then the resulting offspring are going to wind up with the full complement of chromosomes, half provided by the mother, half provided by the father. So how does this understanding of genetics, how does that tie in with this concept of evolutionary theory, this concept of biological change over time? In thinking about this, we have to look at what are sometimes called the forces of evolution. One of these forces of evolution we've already looked at. We've talked about natural selection in some detail and how that works. But you recall we also said that natural selection acts on variation that's present within a species. And so what we need to think about now is the source of that variation. Where is that variation deriving from? Where is it being produced? And we can think about this at 2 levels. We can think about it at the individual level as the source of the new variations that are coming about, and we can think about it at the population level. At the individual level, we'll be thinking about recombination, and we'll be thinking about mutation. At the population level, we'll be thinking about genetic drift and about gene flow. So let's start this discussion talking about recombination. Now, recombination, you recall, is one of those sources of variation at the individual level, and it's the most basic level of where variation is originating. We're going to be talking about humans because this is anthropology after all. So humans have 23 pairs of chromosomes. And this illustration shows what the 23 pairs look like, and you'll notice they're different sizes and shapes, but each member of a pair is about the same size, about the same shape, at least for the first 22 pairs of chromosomes. And what the traits that are coded for on one of those chromosomes in that pair is also coded for on the other chromosome of that pair. So that's how you get your 2 particles, like Mendel said, or your 2 genes for a given trait. You get 1 from each parent. So those 22 that are similar to one another, those are homologous chromosomes. The 23rd pair is not homologous. These are the sex chromosomes, and they're different size and different shape. The X chromosome is much larger than the Y chromosome. And in terms of genotypes, if the individual inherits an X from each parent, that is, if the individual for the sex chromosomes is genotypically XX, then that individual is a female. If the individual inherits an X from one parent and a Y from the other, that is, if it's genetically XY, then that individual is going to be a male. There are some other things that can happen too in recombination. As the DNA is replicated, it can actually exchange segments with the other strand of DNA, creating some new possibilities. But it's estimated that through this process of recombination, that 70,368,744,177,664 genetically unique possibilities could occur. So that's a lot of variation. That gives you a good idea of why no 2 individuals are exactly alike. You got all these possibilities whenever this recombination is occurring. So considering that this recombination is occurring every time a new organism is produced, every time a new child is produced, then this is where most of the variation is coming from that natural selection can act on. So keep that in mind. I'm never going to ask you the 70 trillion number again. You'll never see that again from me. But do keep in mind that recombination is the source of most of the variation that natural selection can act on. Our second source of variation at the individual level is mutation. And what we mean by mutation is a change in the genetic code. There are two basic types of mutation. One of these is point mutation, and what that means is a change at one point, at one location along the DNA strand. Ideally, DNA moves from parent to offspring, just like we were talking about the recombination. But in reality, you can have problems with the DNA breaking. It's a very fragile molecule, and you can have a new base inserted into a location where a different base was originally. And this can modify the amino acid, which in turn can modify the protein. In the case of this illustration on this slide, we've got in the upper part of it a normal red blood cell, nice and round, can carry oxygen very well. That's what it's designed to do. The lower part of the image shows a sickled cell. Now, we're all familiar with sickle cell anemia, and we'll talk about it in more detail a little bit later on. But this sickle cell is caused by a point mutation. At some time in the past, a point mutation occurred that modified the genetic information for those red blood cells, causing this sickling. Now, the sickle cell doesn't transport oxygen as well, doesn't do its job as well as it should, and this causes some health problems. But the point here is that this is caused by a point mutation. The second type of mutation is chromosomal mutation, and you've got a number of different ways that this can happen. You can have a piece of a chromosome breaking off, you can have chromosomes combining, you can have more than one of a pair of chromosomes being passed down, such as you see with Down syndrome. And as you can imagine, this can have a much more significant impact on the individual than a simple point mutation. While mutation can occur in any cell of your body, it's only significant in terms of evolution when it occurs in the sex cells. That is, when it's being passed down from parent to offspring. If you have a mutation that occurs in the body cell, well, that's not going to be passed to your offspring, so that doesn't really matter in terms of the long-reaching effects of evolution. It's only when it occurs in the sex cells, and then it will in turn be passed to your offspring, that it has that significance. But this is where new variation is coming from. When we talked about sickle cell anemia, prior to that point mutation that resulted in the sickled cells, for the red blood cells, you had an HbA allele. But when that point mutation occurred, a new allele was created, the HbS. So while recombination is the source of most of the variation because those new combinations of existing genetic material, mutation is where new variation originates. So this is where those new alleles come from. So while you're thinking about sources of variation at the individual level, just in general, and while you're preparing for the exam, keep in mind that with recombination, that's where most of the variation comes from. With mutation, that is where the new variation comes from. Very important. Keep that in mind. We also need to talk about two sources of variation that natural selection can act on that are at the population level: genetic drift and gene flow. Genetic drift is the first of these. Genetic drift tends to decrease the amount of variation that's available. We define genetic drift as genetic changes in a population that occur as a result of chance factors. So these changes in the population in terms of their gene frequencies, so we don't talk about individual genes when we're talking about populations, we talk about gene frequencies. But when you see these changes in gene frequencies, they're not because of some genetic factor. It's some other non-genetic factor that's causing these shifts in the gene frequencies. Let's look at an example. We'll start with the parent population, and if we're looking at a particular gene, there are two alleles for this gene. One is the capital T and one is the lowercase t. And 80% of the genes in that population for this trait are that uppercase T, that dominant allele. 20% are that recessive allele. And then something happens. Now, usually we think about this as something causing a large segment of the population to be wiped out, some kind of catastrophe. a volcano, an earthquake, a tsunami, something causing part of the population to no longer be able to reproduce offspring. They're no longer available to pass down their genetic information to the next generation. And the result is a remainder population, a surviving population that has less variation because there are fewer people. But also, we see a shift in those gene frequencies. Now remember, the people that died, the people that disappeared, didn't do so because of their genetic makeup. So we can't really predict what the outcome of the gene frequency shifts will be. But just statistically speaking, we can say it's not likely that that remainder population will exactly reflect the genetic makeup of the parent population. So in this example, what we're looking at is that original parent population, which has 80% of the dominant allele, as we said, and 20% of the recessive. And then whatever this cataclysm occurs, whenever this occurs, then we have a remainder population that has only 50% of that dominant allele, 50% of the recessive allele. So you've got a major genetic shift, a major shift in gene frequencies as a result of this non-genetic event. There's a special type of genetic drift that's called founder effect, and with this you don't have people dying. Basically you have people moving away to a new place, and then they're not in contact with that original parent population anymore. Now they're not moving for some genetic reason, they're just moving for some other reason, and as a result they're not taking a representative sample of the parent population's gene frequencies, so their gene frequencies for their new population will be different. We can use our same illustration to talk about this, with our original parent population having 80% dominant and 20% recessive. And then you have that small segment moving away. They're not moving away for some genetic reason, they're just moving away for some other reason. Think of, for example, the Pilgrims moving to New England from their original parent populations in Europe. Now chances are that the gene frequencies of the Pilgrims did not represent the gene frequencies of the original European population. So what we have here, we have our parent population, again with the 80% dominant, the 20% recessive, and then you have a group of people moving away, and just by chance, their gene frequencies are different. And in this case, we're just saying that their gene frequencies are going to be 50% dominant, 50% recessive. The point here is that there's these chance factors occurring that result in a change in gene frequencies. Whether it's the parent population being affected by some catastrophe that kills off a bunch of people, only leaving some survivors with that surviving group having a different gene frequency, or in this case a founder effect, a group moving away but having the same result, reducing variation. Let me give you a specific example of founder effect, and we'll go down to southeastern Africa along the Mozambique-Zimbabwe border. Now, just as we discussed with founder effect, this area was occupied by a small group coming from an originally larger parent population, and they've remained relatively isolated, so they haven't been exchanging genes with other groups. And one of the conditions that you see very prevalent in this population, very common in this population, is syndactyly, this deformation of the feet so that the middle toes aren't developing as fully as the other toes are. Now this trait is relatively rare in the parent population, but when you had this small group move away from the parent population, you may have had one individual that had that trait. But since now you're working with a smaller group, those gene frequencies shifted, where it may have been 1 in 500,000, and I don't know the actual number, so don't quote me on this, but then you have a small group move away. Let's say it's 100 people. Now you've got 1 in 100. So the frequency of that condition is now much higher than it was in that original parent population. And I can give you a little closer look at that. But again, this is caused by founder effect, a small group moving away from a larger parent group for some non-genetic reason. And just by chance, they're not taking a representative sample of the parent population's genes along with them. So this is a form of genetic drift, and it reduces variation. It reduces the variation that's available for natural selection to act on. Now the second source of variation at the population level is gene flow. And as we'll see, gene flow has the effect of increasing variation, increasing the variation that's available for natural selection to act on. So with gene flow, we're looking at populations, in this case labeled with letters, that are scattered across a larger area. And we can conceive of this as over a region, over a continent, over or even across the world. But what's important with gene flow is that the adjacent populations are in contact with one another, that they are exchanging genes, that is, they are interbreeding with one another and producing offspring. So what that means is if a new variation arises in one population, it can be shared with another population. So if a new variation arises in population A, then it can be transferred to population B. And that new variation then can be transferred from population B to populations C and E, in this case, then on to populations D and F. So these populations, these individual populations, don't have to have direct contact with one another for this gene flow to occur. This new variation that occurred in A can spread all the way out to D and F, even though the folks from A may never even see the folks from D and F, but they can spread by this gene flow, by genes flowing from one population to the next. And so what this allows is new variations, beneficial variations that arise, can then spread to a larger group. And so we know that human populations historically have occupied almost the entire globe, and that adjacent populations continue to share genes with one another. So even though before we became highly mobile, more recently in the world, you still had adjacent populations sharing genes. So any new variations that occurred in Africa could be spread to Europe, into Asia, and vice versa. The importance of this for our discussion is that a species occupying a large area, like our species does, can then evolve as a whole. Now you remember back when we talked about Darwin, when you had his common ancestry of similar species, you had a separation between species and then different biological changes accumulated depending on the environment. Well, with gene flow occurring, that species wouldn't separate like that because they're not accumulating different changes. As different changes develop, they can share these that allow the entire species, our species for example, even though we spread out from Africa somewhere between 100,000 and 200,000 years ago, we've managed to evolve as a whole from that time up to the present. And there's a lot of variation in our species, as we can see in this illustration, just a lot of physical variation, but still we're all the same species. We're all Homo sapiens. In terms of evolutionary theory, we do need to talk about just a few more topics, and one of those is coming back to that concept of speciation and how it might occur. Now you remember that Lamarck suggested that one species developed into a new species, transformed. Remember, his was transformational evolution, so he would suggest that species A transformed, evolved into species B, evolved into species C, evolved into species D, just in a straight line. This type of speciation we would call anagenesis. A second model of speciation is cladogenesis. Now, of course, this is reminiscent of Darwin's common ancestry of similar species, where you've got one ancestral species, a separation in populations. That gene flow is not occurring, they're not exchanging genes, and so different biological changes accumulate in each population, so that over time they've separated enough from one another genetically that they're no longer the same species. So you can have a number of species that are descended from an ancestral species. So this is what we call cladogenesis. The other thing we need to think about would be rates of speciation. How quickly might this occur? Well, there are a couple of models here too. The first is phyletic gradualism. And in this model, when we have change on our vertical axis and time on our horizontal axis, it's suggested that this genetic change occurs very gradually over time. So it's a pretty smooth transition, but that's why it's called phyletic gradualism, because it's a gradual change from one species to another. The second model is called punctuated equilibrium, In looking at the fossil record, it seems like at times there are species that exist relatively unchanging for long periods, and then over a relatively short period they change, they transform, they evolve to a new species. So still with our change being our vertical axis and our time being our horizontal axis, what we're talking about is having initially a relatively long period of stasis, or a long period where no change seems to be taking place, and then having a relatively rapid change occurring over a relatively short span of time. Now this change that punctuates our two periods of equilibrium can be thought of as environmental change. So there are new environmental challenges for the species to face. And so only those individuals that have variants, that have traits that allow them to survive in this new changed environment are going to be the ones passing their genes down. So this is natural selection at work. And in terms of the rate of change, we could be talking as short a span as 50,000 years or so. So relatively rapid in terms of evolution, the scale of evolution, in terms of the scale of geology, in terms of the scale of time. That wraps up our discussion of evolutionary theory for now, at least in detail, but we need to keep in mind that what we'll be talking about as we continue will build on those concepts. Next time in Anthropology 1510, we'll be talking about scientific classification, how it developed, and where humans fit into the big picture.